Hello, everyone. Welcome to episode 5 of the NEATCast. And today we're going to look into the topic of how we age in a really amazing study that was led by Dr. Agustin Ibanez's team across 40 countries. And we'll look at the amazing scope and the findings of this study. It is such a pleasure to host Dr. Ibanez today with us. He is a global Brain Health Institute fellow. I followed his work when I was at UCSF, and at that time he was there as well. And now he is at Trinity College. He is also the director of the Global Brain Health Consortium, which is the Latin Brain Health Consortium, and also the world's Red Lat Consortium, which is looking into how we can collaboratively treat dementia. And the paper we're going to look at today was published just last month in Nature Medicine and talks about the exposome of healthy and accelerated aging across 40 countries. So thank you, August, for coming to the NEATCast and talking to us about this very important topic. I would love to, before we dive into the work itself, I would love to hear your journey of how you, as a neuroscientist, started to do this work at this massive scale of collaborating with data across so many countries.
Thank you so much, Jyoti. For me, it's a great pleasure to talk about this research that really has exciting us for trying to develop. For trying to develop a truly tailored precision brain health, I would say. This is the main core of the idea. I come from Latin America, and I have been working with Latin American teams for the last almost 20 years. And one thing that we realized is that, when I was a young researcher, I was trying to fit the results from Latin America to those observed in, the global north, like a way. And it didn't work never. We always have this huge difference in the metrics, in the size, in the dimension of the measures. At the beginning, we were thinking the problem is, poor quality of the metrics, because we have all the MRIs or, not such a good high quality plasma or proteomics metrics, etc. But with the pass of the year, we realized that the problem is much complex. The problem is how we build model of brain health, right? So we create models for the global brain health and dementia using 10% of our population that have a specific admixture and a specific lifestyle and a specific, exposure to environmental factors. And then we do a huge generalization with a kind of quasi-universal models. So this idea started as a frustration, I would say, like being a neuroscientist and trying to understand that the brain is in a body and is in a context. And that if we don't bring this neuroecological framework, neurosyndemic framework, when you have syndemics, from physical environment, from social environment, from whole body health, we are just capturing a piece of the film, so we cannot understand the history beyond that. So that was the first, insight and frustration, I would say, that pushes us to try to think about how develop more, ecological models.
Amazing. And then you started to do this first in Latin America. Tell us the journey of how you started to then have data across so many countries. How did you build that kind of collaboration?
Yeah, so this coming with very early, like 8 years ago, I was blessed to get my first R01 for, integrating different teams in Latin America. Previous to that, I was struggling to try to create a common horizon for different countries and labs. We didn't trust each other. We didn't have any funding. We didn't have any expertise. So we were blind to see that we really have a common horizon. And thanks for the work with Bruce Miller, who was one of my mentors and an incredible researcher with a vision for more global settings, We were able to get our first R1 for Latin America. Then we got another very large grant from the Alzheimer Association. Then we come in with another grant from Rainwater Tau, the Tau Consortium, and then many others. So we start to grow with a very single idea that, um, we can really, I mean, if we want to do precision brain health, we need to understand diversity. Not in the sense like diversity is now discussed in political terms. Diversity is not only about race or gender, it's much more. It's about science. It's not only an ethical problem. It's not only that, we need, we really deserve equal opportunities, but it's also about science. Any model, let's say an analysis of variance or a machine learning or a deep learning, is based in data heterogeneity. So if you have a very coherent non-variable data set, any model will work in a completely different way if you have diversity. And diversity can come in the genes, but can come in the lifestyle, but can come in the brain metrics, in the plasma metrics, in the cognitive cultural. So there are many layers of this multimodal diversity. So we started to create first the Red Lat Consortium. I started to working in Latin America, but then we realized that this is a problem that extends beyond Latin America, also impact Africa or Asia, especially South Asia, right? So we start to collaborate with them. And now we realize that this is a problem that is not only a global south problem. The problem of multimodal heterogeneity is in every place in the world. Maybe in the global south is more accentuated, right? Because we have, more heterogeneity, more multimorbidities, more, social and physical exposure, more accentuated. But this is across the globe, I would say, today, after 10 years working on that. So we started with that, but then we started to work with brain clock. So we published in Nature Medicine a previous paper, already started to showing like, if you come from Latin America, you tend to have an older brain. If you are a female, you tend to have an older brain. But if you have dementia, of course, you will have an older brain. If you have dementia, you are a female and you come from Latin America, you have the oldest brain. So the largest gap between your chronological age and your biological age. That was a shocking result. And we started to look at socioeconomic modulation of the brain clocks, right? And also the pollution, like the impact of environmental pollution on that, and which we observed that it was like a delay. So we start to come up with this idea that how we can create an exposome metrics, like, a metric that combines different sources of environmental sources. But at the same time, we needed something better than brain clocks, because brain clocks are rather limited. You need to have an MRI for each participant. We were thinking in how having more impact, in global settings. So we work with this idea of computational aging clocks, but instead to look at the brain, we start to look at basic whole body, like cardiometabolic health, diabetes, body mass index, hypertension, but also, protective factors, how well you move into the world. That is called functional ability, right? Or how good is your cognition or your education. So we create how good are your sensory abilities. So we create this set of protective and risk factors, and we use them to predict the age of participants. So we created these biobehavioral age gaps. That is the gap between your chronological age and your biobehavioral age that is estimated in the system. This allowed us to have a core metric that is more, easy to get than a brain clock metrics, but also reflect more dimensions of your aging, right? So that was the first step. And then we start to collaborate through the Global Brain Health Institute, that we have more than 300 fellows across the globe. So we contacted the fellows and say, hey, you have data on this, on that, plus the data that we were collecting. So that took about almost 4 years for collecting data, receiving data, harmonizing data. We lost a lot and huge amount of data that was not able to be harmonized across 40 countries. So that's a pity because we couldn't do it. But we were able to create this collaboration between 40 countries and access to also, available data for another thing. So we bring data from many other things, and then we use this exposome level. So we use the standard metrics of physical exposome like pollution that you can get it at country level. Also, uh, the the social exposome metrics like migration, gender inequality, socioeconomic inequality, GDP, etc. And then we have another very interesting thing that is a very, I would say, relevant nowadays, that is political struggle or political instability, right? So measures of democracy, of freedom of party, stability of the democracy, because all these effects, what could be the basic mechanism? Probably is the effect of cumulative stress, right? So in the way that pollution creates inflammation, psychological stress creates inflammation, right? Or socioeconomic inequality or lack of cognitive reserve can also create inflammation. So, or a kind of allostatic overload, like a cumulative burden in response to environmental demands, right? So we bring all together, and the results were really, really amazing.
Yeah, you covered a lot of great things in your statements right now, and it's really impressive work. So let's just pause. You said really, this vision of an exposome that you built, and that term itself is sort of new for the field. Like it was coined around 2005, and really in the neuroscience literature, it's really just been used in the last maybe 5 years or so. And you have been really the leader in this field, bringing this to neuroscience. And you've talked about how the exposome covers not just all the individual health factors like the brain health, the cardiometabolic health, but also the social factors like inequalities and migrations, and then the social political factors like where the person lives and what is their right to vote, their democracy, their political instability. So this combination is extremely impressive and really goes beyond neuroscience. I wonder when you have to get not just body health data, but all of this data across the social political spectrum. You mentioned that you collaborated with the Global Brain Health Fellows. Are many of these social scientists? Is that how you were able to get data on country level social political factors, or are these extracted from larger data sets? I was just intrigued by the level of factors that you got.
Yes, yes. So there are many initiatives that are starting to create, this kind of a huge discussion, I would say, in epidemiology. It's what can be measured for a country or a city as a proper metrics of development, right? So the typical metrics are GDP or the Human Developmental Index or the Human Capital Index, right? We use all these metrics, by the way. But this is a very narrow way to understand what is development, right? So there are many initiatives that have started to measure that. Of course, there are huge limitations for like any other aggregate level metrics. How much you can trust in, the metrics of democracy when a country have some tensions in political issues, for instance. So those are very poor measures. They were not collected at a specific country, but at global level, and has been done by independent group that now is publishing a lot of work with that. But even, with these poor metrics, very poor metrics, and the other thing is there are many multicollinearity. So let's say in general, when there are more socioeconomic inequalities, democracy tends to go down, not always, but tends to go down so they are multicollinearity. So this is why we use a very, robust method for dealing with multicollinearity, but because this is and we control it by the effect of other exposome each time that we were looking to one specific. But no, we was using these country level metrics that are extracted, based on the typical metrics that you can collect also from the WHO or the, other or worrying data sets.
Got it. Very fascinating. And then you talked fast, but I just want to emphasize that with all of these different metrics at various levels, you were able to calculate this or predict the biological age of the individual. And you calculated for then you got these models across all of these 162,000 people that were in the study. And then for each person, you said, what is the gap between their biobehavioral age and their chronological, their actual age, and whether they were younger or older. Their age gap seemed to suggest that they were younger than their chronological age or older than it at that point in time. And then you found that countries that had high GDP, such as mostly in Europe, had better metrics for aging, while Latin America and parts of Asia were somewhere moderate, while parts of Africa were the weakest. They had the most accelerated aging, even though you didn't have so much data from many countries in Africa. They were just in Egypt and South Africa. There was not
Exactly.
multi-country data yet, but at this time there was no U.S. data. So we here hosting the NEATCast and the NEAT Labs in the U.S. At this time, U.S. data wasn't available. Is that right?
Exactly. So first of all, it was a problem so U.S. data, the level of granularity is huge. So the problem is that we wanted to have less granularity to be inclusive of other countries, and we were just focusing on that, and then we didn't include U.S. for any special reason. Honestly, it was because we had to miss so much information from U.S. to harmonize this data that we were focused in the countries that have less data, less evidence, and we managed to work on that. It was just a limitation of our work. We are right now including data from US, so we are doing a lot of analysis, and we are also incorporating other biological metrics in our biobehavioral age gap. It was the first step, and it's a huge gap. It's non-intentional, honestly.
Got it. And so in the new analysis, I mean, I would expect the US to be aligned with where Europe is in terms of GDP and all, but, political situations change all the time. But is that where you would expect it to be?
Yes, but so we don't have strong results yet, honestly, but we see a huge variation in US, and we started to do more specific analysis within US, right? Because the level of inequality, for instance, in US are huge. Even when in Latin America are larger, still in US you have huge level of inequality. You have different, the other thing is the interaction. Usually the people that is living with the poorest socioeconomic conditions are more exposed to air pollution because they live close to the, factories, for instance. So we start to see this pattern, but it's too early to provide robust answer. But definitely, in general, we see similar to Europe, a little bit down, honestly, but a huge variation in US. The other thing is that we have the granularity level that we have in US is really huge, so we can look at much more internal comparisons. Then the other thing that we are starting to doing, we are starting to combine organ clocks and plasma metrics with very exciting results. Then we are starting to incorporate another protective factor. So a paper that will come in Nature Aging probably in 1 month or so is just accepted. We used multilingualims to see how they modulate accelerate aging, and there is a huge effect. And this effect is really robust against, individual socioeconomic variability, other macro level exposomes. So I think that, and that it's a very interesting thing. So the other thing that we didn't leverage in this data set and we are starting to do is lifestyle. So sleep, exercise, nutrition, and we are what the other thing that we are doing right now is using non-linear interactions because the model that we have using in this exposome paper doesn't really capture the nonlinear interactions, capture like the mass effect of the combinations. But when you have like such a huge different factors, like let's say temperature or pollution or water pollution or air pollution, plus socioeconomic inequality, plus political factors, then the regions become more less differentiated because you have such a complex combination of different factors that you shouldn't expect a linear north-south, right, or differences. So we are starting to do nested model with non-linear GAM models, like general additive models that capture one, two, three level of non-linear interactions. And when we look at the high level non-linear interactions, we have a much more complex effect. So we are looking forward to, because this is what we need. We think in this factor as isolated factors. When you think, for instance, I don't know if you are familiar with the risk factors for dementia, for aging and dementia. So there are 14 factors like education or cardiometabolic health. And you can see, how much each factor contributes to the risk. So you can see during childhood, education is a core aspect, right? But this is a very nice analytical way to see the data. In real life, this never happened. If you are poor, you have this all together, and this create a exponential impact. It's not a linear additive factor, right? So this is the other thing that we want to. I think that if we want to move forward in the exposome research, we need to understand the ecological blending of these factors, how they have a huge impact, probably larger impact than considering the contribution of each factor individually. So this is a big challenge. It's for science, it's really hard to measure that. But I think that it's truly needed to develop more precision models.
Yeah, that was one of the questions I had from the paper where you showed that many of these sociopolitical and social factors, but also the individual factors such as cognition and cardiovascular health, and all of these factors are very significantly and with a large effect sizes related to this biobehavioral age gap. But then, like you said, they coalesce together in different individuals. So what would be most important? I think this work gives a lot of insight for countries to think about where do they get the most bang for their buck in terms of programs that can contribute to more healthy aging. And I know you've written about this some in the past as well. You've talked about pro-social interventions. You've talked about interventions at the individual level that take into account aspects of lifestyle, but then also, especially talk about interventions that promote well-being in the context of meaning and purpose, as opposed to following hedonic well-being. And then in a recent commentary, you really were focusing on Colombia and talked about the social political unrest and how that is contributing to the brain health of individuals. So you really have this interest across these levels of how interventions could be conducted, whether it's at the individual level or the systemic social level. So having that vast scope, do you yourself have a specific interest in how you'd want to translate these findings into improving brain health?
Yes, so this is a great point. Thank you for bringing that. So let's do a step by step. At the very short term level, there are many potential translations. So we did country by country analysis of what are the most, robust factors, and there was a huge variation. So that's mean that we can really develop more granular things, trying to don't bring, universal receipts for all, but using local data to create more precision. These simple things is not well assumed and it's not well developed. And this is very basic. We can do, if we go in deep in the local variation of the data, you can develop more predictive models. You can reduce the risk, you can anticipate the risk better. So this is the more basic thing. The other thing is to really combine metrics and having this idea of ecology for being able to, in the future, not in the present days, to develop an individual prediction. But then in the long-term shot, is this very interesting discussion about, individual versus population level intervention. So what we can do at individual level is important, no doubts. So if you change your lifestyle, you have less risk for dementia, that's for sure, right? But there is a problem there. So let's say, take the same people, Juana, who is living in, I don't know, in the one of the richest part of San Diego in US versus Juana living in the most poor way part of Peru. Same person, same original conditions, same genetics conditions. In one case, this person will be, exposed to pollutants, to super fast food. Won't have time for do exercise. Probably will have to travel 3 hours per day to go into work. Versus the same person in the other context, it will have a huge, larger degree of freedom to do behavioral changes. So it will be more easy for that person to have a healthy lifestyle, to have a better nutrition, to sleep well, right? But then it's not just that. Then it's the person living in San Diego will have a less cumulative burden. So a small change in the behavior will have a relatively good effect, because the back is less heavy. Versus in the other condition, you will have to do a very much larger behavioral change to have a quite similar effect on the other side. So this is the thing that if we don't pay attention to these ecological settings, we are creating this very paradoxical recommendation. So the Lancet Commission says, in the global south, let's say in Latin America, you can reduce 56% of dementia, much with preventable risk factors, much more than in the north, which I think is quite the opposite. These effects, these environmental modifiable factors have a huge impact. Of course, when we are doing analytics, you say, oh, okay, if we extract this factor, we can reduce the risk, right? But the people won't be able to do it. That's the point. It won't be, that easy way. So where is that comes the population level interventions? And there are some critical things. We cannot move forward if we don't talk about poverty. If we don't address poverty, of course, we can mitigate a lot of things. We need to do it, but there will be mitigation, and we need to be aware about that, right? So this is one thing. The other thing is we need to start to thinking in more synergetics way. So health is not just medical system, right? So this is the huge problem that we don't understand that we need to bring, socioeconomic settings, the education. We need to bring really transdisciplinary settings for both sides to impact better in the health, but also to understand that the health can contribute to the economy, that the health can contribute to the human capital of a country, right? So this is a more harder thing, how as the countries work, they have a Ministry of Health and a Ministry of Development, and they talk each other, but they develop their own closed plans. And I think that this is a challenge for the population level interventions.
Yeah. So, well, thank you for the telling us about the amazing scope of this study. We're near the end of our time, and I'm just always amazed by the work that comes out of your team and how productive you all are in terms of the different kinds of ways you combine the data. This, as you mentioned, this is almost like a start of a study, and you're looking at these complex nonlinear interactions between all of these different factors. My last question for you would be, as you're so, from your writing, I see you so passionate about this work and really showing how all of these different factors determine our brain health, body health across generations, across cultures, and really looking at the diversity of cultures. But you started as a neuroscientist, and now do you really see yourself being that bridge towards policy as someone that you are are people in the ministries of health, ministries of education, are they seeing the incredible significance of this work to look at how we can improve the health of our populations? And it would be too bad if this stays sort of in the silos of neuroscience and the impressive publications that you have.
No, thank you. Thank you so much. So I think that we are facing now some opportunities. It's not easy at all, but it's not easy at both sides. So my first struggling or fight was try to, as a neuroscientist, I realized that most that we were working in the brain doesn't have any impact in the real life. There is any way to have to do change in real life, that the knowledge that we have about the brain doesn't generalize, doesn't apply to other settings, it cannot be directly connected. So for my first move was, as a neuroscientist, how to bring body and environment to really have an impact, right? But at the same time, the other way around it's really relevant, how we can think in recommendation and in policies that can really impact the health. And I think that this both sides of a same problem, right? So is that this very old-fashioned distinction between biology and culture, we are still, trapped into that way to think. When we think in neurological versus psychiatric disorders, we again think in between more biological or more psychological, this kind of dualistic thinking. I think that I have some hope that we can do something, but at the same time, we are blind to understand complex systems. That's true. So I think that we need to move forward with the hope that in the future we can have more translational impact. But honestly, I think that the translation impact is really small at this stage. That this shouldn't, discourage people to doing that. On the contrary, we want to use science, we want to use biological data to understand society, and we want to understand how social factors are biological embedding, right? We truly believe this. It's just we need better models.
Amazing. Thank you for bringing your passion and your intellect to this really impressive work, really one of a kind in all of the field of brain health and neuroscience. And so thank you, August, for speaking to me today. And it was really significant to host you on the NEATCast.
No, thank you, Joy. It has been a great pleasure. I'm very impressed about how much you know about the study. So thank you so much.